So I have a confession to make. I didn't get the memo that says you have to start your presentation with a "what is this" kind of slide, so I don't have one. The other confession I have to make is I'm a very undisciplined speaker. I've been known to be at the end of meetings and completely throw away my presentation because I thought that there were better questions being asked in the audience and that my presentation would answer, and I find myself in that particular position now. But I don't think I'm going to throw away the entire presentation, but I am going to start with trying to answer some of the questions that have been asked. Now, so one of the questions was around the subject of, when I have an extraction that obviously produces an issue with my medical device, the item that's being extracted, what criteria can I use to disqualify that particular extraction as being overly aggressive? Well, I can tell you that the writers of Part 18 is well aware that the current document is a little bit vague on that subject, and so stay tuned for improvements. But you need to be able to understand that the criterion for disqualifying an extraction is a very clear criterion. If the extraction alters the extractables profile, that's the basis for disqualifying an extraction. So we talked a little bit about physical phenomena like swelling. In my opinion, and I find myself in the rare circumstance that I actually agree with the regulators, swelling will not significantly impact the extractables profile. Swelling won't change the extractables that you find. Swelling won't necessarily change the concentration at which you find them, but it does speed up the extraction, that's for sure. So why would a physical effect like swelling disqualify an extraction? That's why you get no traction with a regulatory agency when you show pictures of my tubing, is 50% the size that it used to be. They'll just look at you and say, well, yeah, that speeds up the extraction. That's a good thing. So you need to be careful that your discussion of disqualifying an extraction solvent is based on a chemical. I have altered my extraction profile by using this extraction solvent. The extractables that I find, because my device has been altered, is different. If you can provide that kind of evidence, then you will most assuredly get buy-in from a regulator that this is an incompatible solvent. So focus on chemistry. Don't focus necessarily on physical phenomenon. We talked a little bit about Part 12. And I can tell you that Part 12 was a very difficult thing for the writers of Part 18 to deal with. But I'm going to give you our opinion, quite frankly. Part 12 describes an extraction process for generating an extract that is intended to be tested biologically. Part 18 is intended to describe a process for generating an extract that is intended to be chemically characterized. And the process of biological characterization and chemical characterization is completely different. The things that make a good extract for biological characterization don't necessarily make a good extract for chemical characterization. And so the writers of Part 18 understood that if the intent of the extract is to produce an article that can be analytically characterized, the process of generating that extract should be optimized for that purpose. And it was the belief of the writers of Part 18 that the extraction conditions contained in Part 12 weren't necessarily optimized to enable analytical chemists to do the job that they needed to do. Secondarily, and I think more importantly from a science perspective, it's a sad truth, but it's the truth nevertheless. There's no person alive that can justify from a scientific basis what the extraction conditions are in Part 12. I'm not saying that they necessarily violate science, but if you ask someone, well, why would you pick 50 degrees for 72 hours versus a higher temperature for a shorter period of time, the only answer you get is, this is the way it's been historically done. The writers of Part 18 said, we're not so much interested in history, we're a little bit more interested in good science. And so the position of Part 18 has always been, you should design an extraction that enables your analytical chemistry, and if that extraction is consistent with Part 12, then that's great. But if that extraction is not consistent with Part 12, that doesn't disqualify it. That doesn't mean it's necessarily bad, okay? With respect to the question about how do I deal with a multitude of unknowns that after I've spent $1 billion, they're still unknowns. I think you're looking for the answer in the wrong place. The answer isn't in chemical characterization. The answer is in biological testing. In a very real sense, chemical testing is a surrogate or a substitute for more expensive and less desirable biological characterization. But if you truly find yourself in the situation where there are numerous major peaks in the chromatogram that remain unknown despite your best efforts, really the only option is to go back and do biological testing. You may find yourself in a situation where you have to do animal testing. It's as simple as that. So again, I don't think you'll find the answer on the chemistry side. The answer is you have to cross over to the biological side. All right, so let's get back to the presentation. The other good news about being the third or fourth speaker is you realize an awful lot of the background information that you had in your early slides you really don't need to talk about, because the audience is at least as intelligent, if not more intelligent, than the presenter. So please don't feel bad that I'm going through the first 5 or 6 slides quickly. I'm just trying to manage my time, and I think my time can be better spent on some of the later concepts. But I've got 2 jobs while I'm standing up here, and my job is to tell you when you need to quantitate and how to do it when you need to do it. It's as simple as that. I mean, if at the end of this presentation I haven't helped you understand that, I've done a really bad job, and Pete will never invite me to come to Leuven again. But realistically, that's what I need to accomplish. And we'll go through. We'll skip the introduction of why is extractables and leachables important, and why is it important to quantitate. But I do want to emphasize a point. We don't just do it from an analytical exercise, because we're analytical chemists and we need to find gainful employment. The fact of the matter is that identification and quantitation are the 2 key pieces of information that enable a toxicologist to do a risk assessment. If you can't give a toxicologist the identity of the compound, then the toxicologist can't go to the literature and eventually calculate a PDE. Okay, so they would be fighting with one hand behind their back if you didn't give them an identity. If you don't give them an accurate quantitation, then they can't calculate patient exposure. They can't calculate a dose. And now you've got a toxicologist with both hands tied behind your back, and I can guarantee you they don't like They don't like being in a situation when both hands are tied behind their back. So it's not an issue of, analytical chemistry dictates that we have to quantify accurately and we have to get the right ID. If we don't do that job as analytical chemists, the whole toxicological risk assessment process grinds to a halt. So that's why it's important. Again, this is all introduction. Okay, so here we are to the AET. The AET is that concept that tells us when we need to quantify. I'm looking out in the audience, and it's a relatively young audience. I remember a time when there wasn't an AET, and it was the responsibility of an analytical chemist to identify and quantify every peak in the chromatogram. So that was a reality. It's not just a nightmare, it was a reality. The introduction of the AET wasn't a gift from analytical chemistry. It was actually a gift from the toxicologists. And the toxicologists didn't think they were doing analytical chemists a favor, but they were. They thought they were just doing themselves a favor. But they actually were enabling the job of the analytical chemist. So when they came to the conclusion that there is a dose of a chemical, any chemical, regardless of identity, that is safe, that there is a safe dose, that again, the most toxic compound, the least toxic compound, it will be safe. Okay? I mean, that's the concept of the TTC. It didn't take an analytical chemist long to know that mathematically I could take micrograms per day, multiply it by the number of days, customize it for my extraction, and all of a sudden that dose-based threshold becomes a concentration-based threshold. And I can use that concentration-based threshold to draw a line on my chromatogram and say everything above has to be identified and quantified, everything below doesn't. Honestly, it doesn't get any simpler. It doesn't get any simpler than that. But just like everything in this world that's simple, it's not that. It's not that simple. You need to understand that in order for the AET to work the way that I just described it, draw the line on the chromatogram, all the compounds have to respond the same in your analytical method. That's a fundamental basis of the utilization of the AET. That in order to draw a line on the chromatogram, all those compounds have to have the same response factor. And unfortunately, it's a practical reality that that's not the case. I mean, I've got it illustrated here, and everybody's got to have their buzzwords. My buzzwords is, this is the response factor illusion. In this particular illusion, each one of those compounds is present in the extract at the same concentration. And that concentration just happens to be the AET. So by the rigorous definition of the AET, each one of those compounds has to be identified and quantified based on concentration. If you were to look at the chromatogram, before you take it into account, the difference in response factors, you would get a completely different picture, and that's the picture that you see in the red bars. Some of the poor responders would have an extremely small peak. You would look at that peak, look at the line of the AET and say, hey, that's a compound that I don't have to worry about. And frankly, obviously that's not true. The concentration plot proves that it's not true. And that's the thing that keeps tax assessors in regulatory agencies up at night. That's exactly what they're worried about, that we are not going to report compounds that need to be reported. So they can't live with this, and neither should we. The question is, well, how do you fix it? How do you fix it? How do you fix this illusion in such a way that those false negatives are revealed? Okay, well, there's one easy way. There's one easy way, and I'm going to propose that way right now. The easy way is to calculate concentrations first. Okay? We're talking about using responses as the judgment for the AET. The AET has always been a concentration threshold, not a response threshold. So let's say, for example, you went through your process for quantitating peaks. Again, I'm not suggesting what that process is. I'm just saying take the entire chromatogram, every peak that you integrate, and quantify it to the best of your ability. And then take that number and compare it to the AET. When you do it that way, your comparison will always be pure. It'll always be accurate because the basis of comparison is an accurate one. It's a concentration versus concentration comparison. Okay, so that's one way. That's one way to fix it. Just do concentrations first. Let's talk about a different way. Because again, I have to tell you, I know what people think when I say quantitate every peak that you can detect in the chromatogram. People say, oh my God, that's a lot of work. Hasn't Dennis ever lived in the real world? Is he just in some kind of an ivory tower? I've lived in the real world for 30 years, so I know what the implications of that is. So there is an alternate. There is an alternate approach, and that's to use response. Go ahead and make the judgment based on response. And then when you've quantified those things that are above the AET, go back and check. Go back and check to make sure that you don't have some really strong responders that looked like they were above the AET on the chromatogram, but when you calculate their concentration, it's now below. It's now below the AET. And take those things off your list. Those things don't belong on your list. So that's a second way to manage that. Why would I advocate for an approach like that? Because I don't like the approach that I'm about to present. And that's the traditional way that we've accounted for response factor variation, and that's using the uncertainty factor. Okay, I've got no objection to the concept. The concept is a good one. The issue is, how do you get that uncertainty factor? And I've got no issue, frankly, with the idea that you get that uncertainty factor from a database of response factors. But the question is how? How do you do it? One of the things we learned when we began to put databases of response factors together is that it's not a Gaussian distribution. Response factors are not very well distributed over the compounds in a database. It's not a normal distribution. So normal statistics don't work. They just don't work. And so yes, I can show an equation where UF is calculated as 1 minus 1 over the RSD, and I can do that calculation. But I have real concerns about the validity of doing that calculation. A, because the distributions aren't normal, and B, because I can't derive this equation. People look at this equation and think that that must be derivable from statistics. That's not the case. You can't get this UF equation by working through statistics. It's just something that years and years ago, many years ago, 20 years ago, before this issue was fully realized, people just dreamed up an equation, wrote it in a PQRI, best demonstrated practice recommendation, and it's just continued on and on and on. So I need to very clearly communicate to you that this is a way to deal with it. It's a way that's accepted with respect to dealing with it, but this is not where the conversation ends. The conversation continues. And again, this is just an example of why I suggest that a concentration-based approach is really the way to manage the AET because it's always 100% right. The only way that it's not right is if your quantitation strategy is no good. You know? I mean, if you use a bad quantitation strategy, obviously this is not going to work. But what I find is that quantitation strategies vary from laboratory to laboratory, but people really do try to do a good job. Okay, again, we've talked a little bit about how do we account for a response factor variation in the AET. Let's talk a little bit about the scenario of, the other one that always comes up is, hey, I can't get to the AET. I've concentrated the heck out of my sample. I've extracted as many devices as I can per unit volume, and the math doesn't work out. It just doesn't work out. My LOD is above my AET, and there's nothing I can do about it. Dennis, what do you suggest we do to avoid the deficiency that we know the FDA is going to give us? Or we've already gotten that deficiency, and what can we do? Well, so I've got good suggestions and bad suggestions. The good suggestions are not the ones in the red. The good suggestions are the ones in the green. Again, it's one of those scenarios that you've already exhausted what analytical chemistry can do for you. So the answer isn't in analytical chemistry. The answer is somewhere else. First answer is make the regulator feel your pain. If you just say, hey, I can't hit the AET, you're not going to get much sympathy. But if in your dossier, you tell the story about, yeah, I can't hit the AET, and these are all the things that I did to try to hit the AET, you're going to get a break from a regulator, at least for trying hard. I'm not going to guarantee that a sob story is going to take you all the way, but it'll get you much further than just a statement, I couldn't get there. So let them know what you did in order to try to achieve the impossible. But I also think that one can make a strong argument that the probability of any of these compounds that show up in the AET gap, that gap between the LOD and the AET, are not likely to be unsafe. And I call that the preponderance of evidence approach. It's the same approach that you would use to convict a killer if you can't find a murder weapon. That doesn't mean that if you can't find a murder weapon, you can't convict somebody of having committed a crime. It just means you need to find another way. And the other way is with a preponderance of evidence. This is all the other evidence that I have that individual committed the crime. Well, this is all the other evidence I may have that would suggest that even though I can't identify and quantify that compound because it's below my LOD, I'm pretty sure that it's safe. Are the materials that you use food contact compliant? Okay. If they are, that implies that they're inherently safe. And the good news is the way you demonstrate that materials are food compliant, you do extraction studies. So it's based on exactly the kind of thing that we're talking about here. Do the materials comply with compendial standards? Again, the reason compendial standards exist is to weed out the bad materials from the good material. It's already a good material if it meets the compendial standards. Are they biocompatible? Do you have 87 and 88, or have you already done some ISO 10993 biological tests? Again, the idea is if it passes these criterion, you already have evidence that it's safe. And if it passes a number of these criteria, what is the likelihood that if the material is food contact, it meets the compendia, it's been demonstrated to be biocompatible, and it's compositionally acceptable by standards such as REACH? What is the likelihood that any extractables associated with that material would be unsafe? It's not zero, but it's got to be pretty close. So build the case. Build the case that the most likely outcome is that those compounds are safe anyway. And then you take your chances. Literally, you take your chances. But I got to tell you, your chances are much better if you've established that you worked hard to hit the AET, and you can provide a preponderance of evidence argument that the compounds are safe even if you can't identify and quantify them. Okay. Again, obviously quantitation is important. If you give a tox assessor a bad number, it's going to be a bad tox assessment. I mean, it's just as simple. It's just as simple as that. And so we focus on the idea that it's the job of the analytical chemist to get as accurate a quantitation as possible. Okay? That's our job. In about 5 slides, I'm going to tell you that's really not your job. But for now, let's assume that's our job. How do we do it? How do we do it? As an analytical chemist, how do we get the most accurate quantitation, where the number that we report is as close to the true value as we possibly can? Well, the first thing that we understand is we remember that this is trace analysis in difficult matrices. So the idea that you would get 100% recovery for every compound is a ridiculous idea. So we just need to remind ourselves with the language that we use that kind of accuracy is not achievable. What kind of accuracy is achievable? Well, I've gone on record to say I believe that extractables and leachables non-target analysis using state-of-the-art chromatographic methods should be plus or minus a factor of 2. Plus or minus a factor of 2 from the true value. That's recoveries of 50% to 200%. If one was to do an experiment and measured spike recoveries, if spike recoveries fell within the range of 50 to 200%, it's my personal opinion, I've obviously argued this opinion with reviewers in that fits the definition of quantitative. But that rubs kind of people a little bit the wrong way because it's really not very quantitative. So we use a term for that. The term for that is semi-quantitative. So we've established that the goal of quantitation in E&L is semi-quantitation. Okay, that's the goal. And we've provided a definition of what semi-quantitation is, recoveries of 50 to 200%. I've been in this business for 30 years. I know that FDA reviewers ask for calibration curves as the gold standard. But, again, let's get out of the ivory tower into reality. If you postulate that there's, 10,000, let's just throw a number out there, 10,000 possible extractables and leachables in the entire universe, who has the time and money to generate 10,000 calibration curves, even if you could find authentic reference standard for all 10,000 compounds? So that's dead on arrival. The idea that we're going to generate a calibration curve for every individual compound is dead on arrival. So we use response factors. So understand this. If your calibration curve is linear, a response factor is essentially a 1-point slope. It's the slope of your calibration curve. And if the calibration curve is linear, it doesn't matter what point on the curve you determine the slope. The slope is always going to be the same. So think of the response factor as the slope. We like to use, when I say we, most analytical chemists in E&L like to use the relative response factor. And you ask yourself, relative versus what? Relative versus an internal standard that's intentionally added to every solution that's analyzed at the same concentration. And the reason we use relative response factors is because it corrects for injection to injection, day to day, those kind of setup issues that would would add uncertainty to quantitation. Again, you can go through and check the validity of these equations. I hope you don't need me to derive them for you. You hear a lot of talk about, well, the way we're going to deal with this is we're not going to generate 100 or 1,000 or 10,000 calibration curves. We're going to generate calibration curves for surrogates. We touched maybe a little bit about this on a previous question. I'm going to link my analyte to a surrogate that I have reason to believe it has the same response characteristics as my analyte. And I'm going to use the calibration curve for the surrogate to make my quantitation accurate. Great concept. The issue is, how do you make that linkage? How do you make that linkage? Well, I can tell you that retention times were proposed as a way of making that linkage. In other words, all the compounds within a retention time window would have the same response factors, and that doesn't work. That's not the case. It just doesn't work. There is the suspicion that you should be able to link structure to response. And I think that's a true suspicion, but nobody has figured out how to do that. If it's a simple correlation like, hey, my analyte is a phthalate, so I'm just going to pick a phthalate as a surrogate, that doesn't work. The response factors of phthalates is all over the place. So this is a great concept, but practically speaking, no one has ever been able to show me a way to pick a surrogate that's not voodoo, okay, for lack of a better word. Yes. And this is the point where I wanted to get to in my presentation, and it's thinking a little bit different. I've got a reputation for thinking a little bit different, and it served me well and got me in trouble. But in this particular case, we as analytical chemists, we're trained to develop methods that are accurate. It would go against our instinct to develop methods that aren't necessarily accurate, but I'm going to suggest to you that in the context of tox assessment, our objective isn't to make the concentration that we report accurate, it's to make it protective. And what I mean by protective is that when we report a concentration to a toxicologist, we should be able to look that toxicologist in the eye and say, the concentration that I'm reporting, there's no way in the world that the actual concentration in the extract is higher than that. So you can be confident when you do your tox assessment on my number that there's no way that you could conclude the compound is safe when in fact it's unsafe. Okay, so think for a minute that our objective as an analytical chemist becomes to produce a concentration that's protective and not necessarily a concentration that's accurate. What this looks like is a picture. But our criteria now isn't that recoveries be 80 to 120% or 50 to 200%. Our requirement now is that the recoveries always be greater than 100%. Okay, so that's the new challenge to analytical chemists from a quantitation perspective, is that you should be developing quantitation methodologies that ensure that the concentration that you report is always 100% or greater. All right? And I think that if you would think about that, the way that you produce quantitations that are protective is slightly different than the way that you would produce quantitations that need to be accurate. And thank you for your time.